Testing. Good morning, everyone. We are the 2016 TAS Taipei iGEM team. My name is Austin, and our project is called Counteract. And what we're trying to do is to prevent and treat cataracts using non-invasive methods. So the issue that we're dealing with here is cataract. And cataract is a serious problem when we look at common diseases around the world. First, cataracts are the #1 cause of blindness. It constitutes about 48% of the world's blindness, and a little bit of math gives us over 20 million people who are suffering from cataracts. And so it's certainly a global issue that needs to be dealt with. But first, what exactly are cataracts? Well, in short, cataracts are the clouding of the lens. As you can see in the diagram here, the lens, as shown in blue, is the structure in our eyes located behind the cornea. And the thing about the lens is, it is made up of tiny lens cells. And these lens cells mainly contain what we call crystalline proteins. It's a type of protein. And the crystalline proteins is what keeps the lens clear and is what allows light to pass through. So how we see is light will pass through from the cornea, through the lens as facilitated by the crystalline proteins. It will hit the back wall of our eye, and that's how we see. So if we have a healthy lens with normal crystalline proteins, what we have is a normal eye, and you would expect the vision provided by this eye to be rather optimal, right? But what happens when the crystalline proteins are degraded is that we would have a cloudy lens, and you can see in the center here, there's an opaque region. That's because of the cloudy lens, which in turn is because of the degraded crystalline proteins. So you would expect the vision provided by this eye to be blurry. And this blurriness of vision definitely does affect everyday activities. So to have a better understanding of how exactly and why exactly do cataracts form, we need to know a little bit more about crystallines themselves. And previously, just now, we learned that the crystalline proteins are what keeps the lens clear. But perhaps the most important point about the crystalline proteins with respect to the formation of cataracts is that the crystalline proteins must remain functional for our entire lives in order for us to see clearly. And that's because when the crystalline proteins in our lenses get degraded, they will not get replaced. They don't get replaced by newly generated proteins. And that's as opposed to when most other proteins in our bodies do get replaced. They do get renewed by newly produced versions of the proteins. So if we compare, to illustrate this point, let's compare, say, a 5-year-old kid versus a 65-year-old man, and let's compare their protein makeup. So just as an example, let's look at their muscle protein. If we look at the muscle protein in both individuals, you would actually expect the age of the muscle protein in both of them to be around the same. It would be around 1 month old. And that's because the muscle proteins would actually be renewed. It would actually renew itself for every 1 month. So that's interesting because if we go back to the crystalline proteins, the age of the crystalline proteins in the 5-year-old kid would be 5 years old, while the age of the crystalline proteins in the 65-year-old man would be 65 years old. What that entails is the crystallines in the lenses of the man must remain functional for all 65 years. So through scientific literature, we found 5 main causes of cataracts, 5 main factors that has been found to be causing cataracts. And first, we found that diabetic patients have a higher risk of developing cataracts. We also found that long-term exposure of our eyes to UV radiation can lead to the onset of this disease. And so will the injury. Certain types of injuries to our eye increase the risk of developing cataracts. And we found some children who develop cataracts right after they were born simply because of genetic mutations. And we call this type of cataracts congenital cataracts. But by far the single most prevalent cause of cataracts is old age. Simply, as time goes on, as we age, all sorts of damage build up in different forms. They accumulate to cause cataracts. And this fact is shown by the pie chart here, which shows all the people who have been diagnosed with cataracts. You can see an overwhelming majority, right, of all the people who have been diagnosed with cataracts are either 45 years old or above. So there's a clear correlation between old age and the onset of cataracts. But regardless of what is causing cataracts, today there's only one method of treatment that is globally recognized, and that is cataract surgery. And every year around the world, there are many people who undergo cataract surgery, but it comes with various different problems that we can avoid. First of all, cataract surgery is inherently costly. Let's take US for example. In the United States, if you perform a single cataract surgery procedure on one single eye, it costs over $4,000 US. And if you consider the fact that most people develop cataracts in both eyes at a time, that adds up to a huge amount that the patients will have to then pay for. So secondly, cataract surgeries, to perform cataract surgery, you need trained surgeon, right? You need proper facilities, proper tools, like a proper operation room. But the thing is, in most developing countries and most other countries, they don't have as many of these facilities and resources as they would like. So what happens because of the costliness and because of the various different requirements is that patients in developing countries or simply patients at a lower financial status would not be able to access cataract surgery as a method of treatment. And that's definitely an issue that needs to be resolved. Finally, cataract surgery, like all other surgeries, takes on an invasive approach. What that means is when you're doing cataract surgery, you're cutting into the person's eye and you're removing the lens. As shown by the diagram here, you're removing the cataractous lens and replacing it with an artificial lens. And this approach of cutting into the eye comes with inevitable risks that we can avoid by developing non-invasive methods that are all the while more effective. So this is why our goal is using non-invasive methods to not only treat cataracts, but also to prevent cataracts from forming in the first place. So for the rest of the presentation, we are going to talk about the experimental part of our project, how we are experimentally developing and testing new ways to prevent cataract formation and treat cataracts. Then we are going to talk about the prototype aspect of our project. What are the non-invasive and affordable ways that we plan to use to deliver our final product, deliver our final drug into our eyes efficiently in a way that everyone around the world can access this as a method of treatment. Then, finally, we're going to share what we've done and what we plan to do for policy and practice. First, how we have used research to shape the direction of our project. Then, how we are going to use education and outreach to not only raise awareness about cataracts as a global issue, but also raise interest, right? Raise interest in synthetic biology as a new and emerging field of study. So, without further ado, we are going to jump right into the experimental part of our project, and Fiona will take it from here.
So I'll be covering the experiment part of our project, which can be broken down into two sections. First, in prevention, we want to prevent the degradation of the clumping of the proteins, so there will be no cataract formation. Second, in treatment, we want to break apart the proteins that are clumped together in order to reverse the cataract formation. Degradation of the protein happened because of something called hydrogen peroxide. Hydrogen peroxide is a natural byproduct of metabolism in the cell. In our lens, when we have too much hydrogen peroxide, it leads to protein misfolding, breaking apart, and clumping. The damage of hydrogen peroxide on these proteins is what we call oxidative damage. Antioxidant provides a defense system against these oxidative damage by detoxifying hydrogen peroxide. So, when we have more antioxidants than hydrogen peroxide, the proteins can still function normally. However, when hydrogen peroxide overwhelms the antioxidant defense system, it leads to protein damage. The main antioxidant in our body is called glutathione, or GSH for short. New cells in our lens form around the outer layer of our lens, and as they form, they push the older cells to the center of the nucleus. In older cells, we have less GSH, and so when hydrogen peroxide overwhelms the defense system, it leads to more damage in the proteins. This picture illustrates GSR's role in this defense system. GSH is able to detoxify hydrogen peroxide because as hydrogen peroxide oxidizes GSH, it is converted to water and oxygen molecule, so it can't inflict oxidative damage on other proteins. GSH is converted to GSSG when it's oxidized, and GSSG can be recycled back to GSH with the help of GSR enzyme. So in our DNA construct, we want to constitutively express GSR protein here in order to increase the efficiency of recycling. These two animations show GSR in action. So in this normal lens, you can see the two pink dots or circles are GSH antioxidant. And GSH is able to prevent H2O2 from oxidizing the crystalline proteins. GSR enzyme is able to recycle GSSG, which is GSH together, back to GSH. On the other hand, this is a cataractous lens. Without GSR, GSSG is unable to be converted back to GSH. So the hydrogen peroxide is able to directly oxidize the crystalline proteins and hence breaking them apart. So that's the prevention part of our project. And in treatment, we want to disaggregate the proteins that are already clumped together by restoring the solubility. Through literature research, we found a compound called 25-hydroxycholesterol, or 25-HC for short, which does exactly the job we want to do in treatment. The precursor for 25-HC is called cholesterol. It is a naturally abundant steroid in our body. It can be converted to 25-HC by the help of CH25 enzyme. So in our DNA construct, we want to express this protein in order to increase the amount of 25-HC we have in our body. In order to test our two constructs in prevention and treatment, we want to create an environment that is similar to cataract lenses. So what we first did was we dissected fish, isolated their lens protein, and dissolved them in a solution. We then added different concentration of hydrogen peroxide, which we expect to lead to protein clumping and creating a solution that is more cloudy than the one without hydrogen peroxide. We decided to use the lens of Priencanthus mercancanthus, a freshwater fish because of its availability. So in here we dissected the fish and we isolated the cortex and nuclear protein. We defined cortex protein as the ones with the newer cells at the outer layer of the lens, and the nuclear proteins to be the one in the center where the older cells are. We put these proteins in solution form and shook them overnight in order to dissolve the proteins. So we got our solution, and we wanted to confirm that whether or not the proteins that we're looking for are actually in the solution. So we first ran a burette test. The purple color here indicates that there are indeed protein present in the solution. We then ran a protein gel. The purpose of running a protein gel is it separate different protein by their molecular size. The smaller the protein is, the further it runs down the gel. The three most abundant protein that we're looking for is alpha, beta, and gamma crystalline. So we matched the size of the protein with the band, with the location of the band, and we did find alpha, beta, and gamma crystalline. So after confirming the protein that we're looking for are actually in the solution, we added different concentration of hydrogen peroxide. We then measured the absorbance value, which is reflective of how cloudy the lenses are. This is Dennis, the face of iGEM 2016, measuring the absorbance value using the spectrophotometer. These are the graphs we got from our cataracts model. And from these two graphs, we infer that hydrogen peroxide has a more profound effect on the nuclear protein than it has on the cortex protein. This is expected because nuclear protein is where all the older cells are. So with less GSH, it is more vulnerable to oxidative damage. To sum up the experiment part of our project, in prevention, we want to create GSR because it will increase the efficiency of the recycling of GSSG back to GSH, which is the antioxidant in our lens. In treatment, we want to create CH25H in order to create 25HC, in order to restore solubility to the lens and disaggregate the proteins. In cataracts model, we simulated an environment that is similar to cataract lens in order to test our prevention and treatment construct. We concluded that from now on, we will focus on the nuclear protein, and we also need to try different buffers for controls and run more trials to determine the optimal concentration for hydrogen peroxide. So that concludes the experiment part of our project, and I will now pass on to Pedro.
Hi, so now I'm going to go over the prototype of our project. So this is where we apply the research and experiment into real-life applications. So for us, we want to deliver the protein that can prevent and treat cataracts into our lenses. So this year, we designed a non-invasive prototype that can not only prevent bacteria from entering the body, but also directly delivering the protein into lenses. So how exactly will we do that? Well, according to our survey, most people don't want foreign bacteria in their body during medical treatment. Well, bacteria produces the protein that we want, but we don't want the bacteria in our body. So we need to figure out a way to isolate and extract the protein from the bacteria. And this process is called protein purification. So as you can see here, we inserted a his tag into each of our constructs. This his tag allows us to tag and isolate the protein produced by the specific constructs. And with protein purification, we can prevent direct contact between human body and bacteria. So now that we have the purified protein, we need to figure out a way to deliver the protein into our lenses. And the two non-invasive methods that we came up with are contact lenses and eye drop. So for contact lenses, we want to attach protein on the interior surface of the contact lenses. And for eye drop, it will contain the protein solution that we will make. So even though these two non-invasive methods are relatively simple to apply, we're not sure if the protein can actually deliver into our lenses. So in order to elaborate this, the main issue we're facing right now is that, as you can see here, the cornea is the outer surface of our eye, and it has two layers, it contains two layers. So it's usually difficult for substances to pass through the layers into our lenses. So after some literature research, we found a specific type of nanoparticle that is not only biodegradable, but can enhance the protein delivery into our lenses. So as you can see here, the biodegradable nanoparticle encapsulates the protein, and it passes through the first layer of the cornea. And once it passes through the cornea, it biodegrades and releases the protein into our lenses. So this is an SEM image of the nanoparticles that we made. Right now, the nanoparticles do not contain proteins, but we confirm that they are the right size that we want. So with our prototype, we develop non-invasive methods to prevent bacteria from entering the body and also deliver the protein into our lenses. So now that we have a prototype design, we still have many unanswered questions, such as how fast does the biodegradable nanoparticles actually degrade? Or how much hydrogen peroxide do we need in order to have cataracts? Well, because these questions can't be answered with experiments, we have to turn to computer modeling. Computer modeling is when we use mathematical calculations or computer simulations to predict experimental data. So here's a flowchart of the summary of our modeling for this year. So one main question that we have is that how much protein can each nanoparticle actually contain? So in order to answer this question, we made two models. The first one is the nanoparticle concentration, meaning how much nanoparticles do we make, and how much do we have to send into our lenses. And the second model, as you can see on the blue portion, is how much protein each nanoparticles can contain. So with these models, we can figure out specific values for our experiments. This is an example of the model we have so far, and this answers how the hydrogen peroxide affects the GSH activity. As you can see in the graph, the higher the concentration of the hydrogen peroxide, the lower the GSH activity. So with these models, our experiments can run more smoothly. So now that I covered the prototype and the modeling of our project, I will hand my microphone to Zach.
Hello, me along with Angela are in charge of the policy and practice of our iGEM team this year. And policy and practice encompasses a number of things, but mostly what it has to do with is the importance of our project, the effects of it in the community, and how it's perceived in the community. So what we're trying to do is we're trying to create a viable project that people perceive to be helpful and that also is actually helpful. So we take some data on what people believe, what people think about the directions that our project are going, among other things. So the first aspect of our policy and practice is research, and that includes research on public opinion from doctors and vets, and then we use that data to create a marketing plan for our actual prototype. And so the first way that we gathered public opinion was through a bioethics panel, which was in this very room not too long ago. And what was useful about the bioethics panel is that we got a lot of good information from experts in various different fields. So it's not like we were just talking to scientists or just talking to researchers or doctors. We were talking to people who were also, English teachers. And so that gave us a good wide range of information as to what their opinions were. And one of the major things that we gathered from that was that the appeal of our current project is its relative ease of access, how easy it is to apply, and then also how inexpensive it is. And so that was very useful. But then another thing that we did to get even a wider range of opinions was through a survey, which we sent out to our parents and our grandparents and our peers, in which we got people to fill out at the spring fair. And we got a lot of really interesting information, which has affected our policies in the development of our prototype. And one of those is that very few people know about cataracts, but a lot of people know people who have had cataracts. So that shows that it is an important issue, but that a lot of people need to be educated about it. And I'll get to that later, but that has more to do with the outreach of our project. And so some more statistics that have directly affected what we know about it is that a lot of people don't want to pay for cataract surgery. And that's partially because, as we mentioned, it's very expensive, but that's an interesting statistic considering our community and how we tend to be fairly well off, and yet people still don't want to pay for cataract surgery because it is very expensive no matter who you are. And then another interesting fact is that even less people want to pay for cataract surgery for their animals. And that's a recent direction that our project has taken in terms of, can these proteins be applied to pets? Because many pets, such as dogs and cats that have similar lenses to us, are equally exposed to UV radiation and develop cataracts with age. And so that's an interesting part of where our project can develop. And another statistic that isn't shown is that 75% of people who took our survey said they'd be willing to take eye drops to prevent cataracts. So as of right now, that shines out eye drops as the most promising direction for our delivery mechanism, which is interesting. And then so the other thing that we have to do is we have to talk to people who are actually delivering the surgery, because these are the people on the front lines. And so we got a lot of interesting information about how cataracts is delivered and how long it takes. And it's a fairly quick and easy process, but then the problem is, of course, the problems that come afterwards. And so we got a lot of useful information about that, and that was interesting. And then so we use a lot of this data and we pull it together to develop a marketing plan, and that includes how our project is affecting the real world and what are the benefits, and then also why is it better than the current methods. We need to make sure that we're constantly evaluating our project as it's compared to cataract surgery, the only current method. And so here are some generalized ideas that we took into account while we were developing our marketing plan. And it's still in the developmental process because we're still gathering data from people, and we're still doing a number of things relating to our research and human practice. And then the other aspect of our human practice is education. We need to not only spread awareness about cataracts, but also just raise interest in science and synthetic biology in our project. And so we started off with an NYMU summer camp, which is where we learned quite a bit about synthetic biology and science. And then we took the next step and started teaching other people about it. And we started off very, very young with kindergartners. And so we started off with very simplistic scientific ideas, but they had an incredibly complex understanding of these ideas. They were very interested in it, and I think it was very successful in getting them interested in science. And we just did some nice experiments with electricity and light and explained to them a few concepts. And then we had a bit more background information with the 7th graders because they already knew about DNA, they already knew about base pairs, so we began to explain to them what we do in the synthetic biology class. They ran a few gels, we got to show them how things work, and I think they enjoyed it as well. And then with high schoolers for the IBSL class, they had even more background information, so we got into the specifics of cataracts and how our project is working, where we're going with it, and that was also very successful. And then so another aspect that is pretty distinct from education, but that is very much related, is outreach. And that has to do with raising awareness about our project, raising money to send to charities that are putting work into the same work that we're doing. And so with outreach, one of the first things that we did is we started fundraising. And that was through the bake sale, which some of you might have seen, where we sold eyeball cupcakes, because that's where cataracts happen. And we also do a number of other things, like we had a spring fair, which was partially to raise money. We had a donation box, and also to raise awareness. We had a small little demo to explain how cataracts works. And I think that they enjoyed it, and that's Sage, our biggest fan, spending time with us. And one of the things we've taken up more recently, which we think is very promising, is the development of a National Cataracts Day. So we're going to start a dialogue with some US senators pretty soon and talk about the development of a National Cataracts Awareness Day. And what this day is going to be is it's just going to be a day dedicated to the awareness of cataracts. But it's more beyond that, because we hope the goal of this day is not only to raise awareness in the general public and to help them realize that this disease is relatively preventable if you take the right steps and you're aware of the fact that you might be genetically predisposed, or you need to take better care of your eyes and stuff like that, and find it at earlier stages. And so there's raising awareness in that respect, but then there's also raising awareness in terms of raising money for research in cataract surgery, and also just getting more people interested in solving this problem like we are. And so we're starting a dialogue with senators pretty soon. We're going to send out to multiple, and hopefully one of them will respond, and we'll see what happens. But one of our main arguments is that'll hopefully appeal to these senators is that seeing is a natural human right, and we need to make sure that everyone has that, even no matter how old they are. And so we're going to be sending that out pretty soon and seeing what we can gather from that. And that's pretty much the gist of our human practice, policy and practice. We wanted to thank you all for coming out, and we appreciate you being interested in our project. If you are interested in this kind of stuff, there's a lot going on after school. At about 3:50 in here and several rooms around, there's going to be Science Research Symposium. And so, yeah, and if you're finished with the surveys, you can hand them to our representatives, which will be in the back pretty soon, and thank you for coming.
And we'd like to thank Mrs. Cruz for helping us with the modeling slide, and
Yes.
also Mr. Brundage for the National Cataracts Day, Awareness Day.